Well, dear colleagues, friends, and all dear ladies and gentlemen, it's a great pleasure for me today to award Dr. Christopher J.L. Murray the honor of the DGHM Lecturer of the year 2022. And I send my greetings to Chris, who's already online. So technology is working today. You will listen to a fantastic talk. I'm absolutely convinced about this, and thank you for coming. We will award him for his outstanding contribution to global health by initiating the Global Burden of Disease collaboration, his fantastic work towards tackling antimicrobial resistance, AMR, and analyzing the performance of healthcare systems during the COVID-19 pandemic. The DGHM Lecturer Award is presented to internationally renowned professionals from the fields of infectious disease, research, immunology, or hygiene. This year, I'm extremely happy to announce Dr. Christopher Murray for this prestigious award. Let me briefly introduce Dr. Murray to you. Christopher Murray is an American physician, health economist, and global health researcher with a long and outstanding career. He is a professor at the University of Washington in Seattle, where he is chair of health metrics science and the director of the Institute for Health Metrics and Evaluation, the IHME. Beginning in 1919, he has worked on ways to measure the burden of disease and disability around the globe. Along with medical demographer Alan Lopez, he developed the disability-adjusted life years, this fantastic concept, to measuring the global burden of disease. He has led several projects to gather that data, disease by disease and country by country. And I think we can only appreciate this even more after this pandemic that we're all experiencing here, how hard this is to do. The aim of these efforts, which involves the work of hundreds of researchers, is to provide data for policymakers around the world to allocate healthcare resources. Murray previously served as director of the Harvard Initiative for Global Health and as executive director of the Evidence Information for Policy Cluster at the World Health Organization, WHO. He graduated from Harvard University in 1984 and was a Rhodes Scholar attending Oxford University, where he earned a Doctor of Philosophy in International Health Economics. In 1988, he returned to Harvard, where he specialized in internal medicine and earned a medical doctorate. He was appointed various professorships and several prestigious awards working on measurement of health and health outcomes. He was also part of the Disease Control Priorities Project, and from 2005 to 2007, Chris was director of the Harvard Center for Population and Developmental Studies. In 2007, the Bill and Melinda Gates Foundation, along with the state of Washington, established the IHME and selected Murray as its leader. As head of IHME, Murray greatly expanded on his earlier research, leading on efforts by 486 researchers from 302 institutions in 50 countries to produce global burden of disease reports, which are continuously increasing in data wealth. To give you an example, in 1990, for example, researchers cataloged 107 diseases and injuries. The 2013 report involved creating and then analyzing a database of over 800 million deaths and produced estimates for death and disability from 240 health conditions. Chris, it is indeed an honor that you're here today with us, although it's only virtually, to accept this award. With your vision, you have brought meaning to public and global health research, in particular when you initiated the Global Burden of Disease study. Most recently, you published for the first time the global burden due to AMR. Your research brings more meaning to the fight against AMR, so the silent pandemic that we are watching and analyzing since decades. I'm extremely honored that the Robert Koch Institute, the National Public Health Institute of Germany, could participate in this significant research, and I thank you for all your determination and efforts. On behalf of the DGHM and everyone sitting here, I would like to thank you for your contribution to public and global health research, especially for initiating the concept of DALIs, leading to global burden of disease study. Our heartiest congratulations to you, and we are delighted that you have accepted our invitation to give this year's DGHM lecture. And I will now virtually present you with a certificate. It should be, here you see it also on the screen, but I will read it to the audience and to you. The Deutsche Gesellschaft für Hygiene und Mikrobiologie, DGHM, honors Professor Christopher J. L. Murray as the DGHM Lecturer 2022 for his outstanding contribution to public health worldwide, in particular through initiating the global burden of disease collaboration, tackling antimicrobial resistance, AMR, and the assessment of the performance of healthcare system during the COVID-19 pandemic. The 74th annual meeting of the DGHM, Berlin, September 6, 2022. Professor Dr. Georg Hecker, President of DGHM. So now, Chris, the floor is yours.
Thank you very much. It's a great honor. I'm sorry I'm not there in person, but hopefully the technology will work. And again, thank you not only for the award, but also for those very kind opening remarks. Those were very generous. I am going to switch to my slides. I hope they come through. There they are. Okay, so what I'm going to do is walk through this extension of the global burden of disease work that was nicely described. This is a body of work that in various guises has now been going on for more than 3 decades. There's actually, I think, an interesting retrospective about 3 decades on the global burden of disease effort that will be in Nature Medicine next month, that tries to trace a little bit of what's happened over 30 years. But about 5 years ago, Sally Davies, who is a great champion of AMR, as I'm sure you are all aware, and who was a board member for the Institute for Health Metrics and Evaluation, challenged us to extend the GBD work to encompass AMR. And the result of that work, and quite a broad collaboration, anchored both at the Institute for Health Metrics and Evaluation and at the Big Data Institute at the University of Oxford, but with many people participating, was this paper in January in The Lancet. And what I will try to do is walk through how we did the analysis and why, as I'm sure this audience is very familiar, it turns out to be quite complicated, both because it's a complicated subject with many different drugs and pathogens, but also because of the patchy nature of the data. So what we published was an analysis for 23 bacterial pathogens and 88 drug bug combinations for which we found at least enough data or some data. And the basis of this analysis, right from the beginning, we recognized that there were 2 different analyses that we needed to do. The first was the more traditional analysis in the AMR space, which is to look at the number of infections and hospitalizations and deaths where drug resistance was present, and that we call associated with drug resistance. So both deaths and DALIs. We also have incidence and prevalence. And then the second analysis was more in keeping with how the rest of the global burden of disease work is done, and that is to try to isolate the incremental effect of having a resistant pathogen compared to a drug-sensitive pathogen. And that is what we call the burden attributable to AMR. Now, the reason we did both is that there is good arguments in the literature, think about MRSA compared to MSSA, that say perhaps drug-resistant pathogens are not substituting for drug-sensitive pathogens. And for that reason, we did the entire study using both of these counterfactual definitions. Now, this causes some confusion with some users, but of course, for this audience, it will not. But it has been an interesting challenge to communicate both constructs to the media. Now, it would have been a very easy study to do if there was many studies published and data sets available that directly counted death or some other outcome due to with the presence of a drug-resistant pathogen. But in fact, that type of data that goes from the individual patient and their clinical diagnosis right through to outcome with appropriate microbiological documentation is extremely rare. And so we ended up having to construct, as we often do in the burden of disease work, a much more complicated approach to estimation, but one that allowed us to use many more data sources from around the world to try to get at different aspects of drug resistance. Now, this flow diagram summarizes what took a number of years for us to come up with how to do this. And I'm not going to go through the flow diagram, but I will go through the 9 steps here very briefly, and then talk about the 10th step, which is just bringing all the numbers together with appropriate documentation of uncertainty. So, first 2 steps. The first step in the analysis was to estimate the number of infection-related deaths by country, by age, and by sex. Now, you would think that this might be simple, but it is much more complicated than you would think, because here, to understand AMR, we wanted not only to understand what in medical statistics is known as the underlying cause of death, following the WHO principles of the International Classification of Diseases and Injuries, but we wanted to know, regardless of your underlying cause of death, was major infection on the pathway to death. So think about the diabetic who dies in the ICU from gram-negative sepsis. We wanted to know how many people were in that category because AMR may be playing a role there, and in addition to the people whose underlying cause of death might be a pneumonia or a urinary tract infection or a bloodstream infection. And so we did that analysis. It was separately published in the Lancet a few years back, and then we've kept it up to date. And this little table on the right just points out how at the global level, there are many deaths that from non-communicable diseases where the underlying cause is, let's say, diabetes or some other process, a heart attack, where the patient actually dies from a major infection. 5 million plus, in fact. So that was an important step in the analysis, and a smaller fraction, 5% of injury deaths, the final pathway to death is also through major infection. Now, the basis for this analysis is the small set of countries in the world where we were able to obtain multiple cause of death data, and then we also supplemented that with hospital discharge data where we get multiple causes of both admission and discharge. The second step in the analysis was to figure out amongst the deaths that are from a infection or where infection is on the pathway to death, what is the clinical infectious syndrome that that patient had. So in other words, I might have been admitted with diabetes and ended up in the ICU with gram-negative sepsis, but was my infectious syndrome on that route a bloodstream infection, or was it a UTI, or was it a pneumonia, or an abdominal infection, et cetera? And again, we used hospital and multiple cause of death data to try to figure that out statistically. Step 3, which is really quite critical, is to figure out the case fatality ratios for each infectious syndrome and each pathogen. Now, case fatality ratios vary by age, They vary by country because of access to antibiotics and to other interventions and supportive care. And so we systematically reviewed the published studies and found other large data sets where we could try to estimate the case fatality rates. And then we looked statistically to see how case fatality rates varied by level of healthcare access and quality. So the global burden of disease produces an index that we used as a predictor in this case that tells us about the, roughly speaking, the access to healthcare and the quality of that healthcare. Now, I think there's a lot more work to be done on the case fatality part. Case fatality rates vary dramatically across pathogen, across infectious syndrome, and across country. So that is an area where I think in the future, as we look towards improving this body of evidence, there's a lot to be done. Number 4 in this analysis is interesting in its own right. We have several publications coming just on this part as a follow-up to this study on AMR, and that is to figure out for each infectious syndrome by age, by country, what is the distribution of the pathogens causing that infectious syndrome. And of course, that distribution of pathogens is changing. So think about in the era of COVID and the widespread use of glucocorticoids, that we've seen a rise in many parts of the world of fungal infections. And so the pathogen distribution for a particular infectious syndrome, let's say pneumonia, is changing. So this task of estimating the infectious syndromes is very important. It's important both for clinical management, but also for the development of new vaccines and drugs. And I'm actually happy to say that some of the companies in the pharmaceutical industry have already acted on our AMR analysis to reprioritize research on new interventions or new drugs for certain pathogens that I'll come to. So expect more on this step 4 to be in the literature coming soon. Step 5 in our analysis was to use sales data from groups like IQVIA, as well as survey data on access to antibiotics to try to estimate in low, middle, and high-income countries access to antibiotics as a key variable that goes into our step 6, which is modeling the prevalence of resistance. So you might have thought that, the biggest task in this analysis was just the prevalence of resistance. And of course, that is a critical task, and that's step 6 here. But in reality, this perhaps was the one that was the more straightforward analytically. But of course, it's driven entirely by access to sensible data. And there are a combination, a big part of our 5-year project was trying to negotiate with data holders around the world access to their microbiological data so it could go into this global analysis. We ended up finding data only for about 50 million isolates to analyze the proportion, and that's supplemented with other sources that you'll see, so it gets larger than that number. But that's sort of 50 million worth of detailed microbiology. And then we used, a geospatial statistical modeling framework to estimate the prevalence of resistance. And that would be fine if we wanted to stop at the incidence, prevalence, death, burden of disease that is associated with drug resistance. But we wanted to go that further step and figure out in a more minimalist sense, what is directly attributable to resistance compared to having a drug-sensitive infection. To do that, we also have to know about the correlation of resistance, because otherwise we would end up double counting, because as we all know, there's quite a significant correlation across some of the resistance patterns. And there we had to model out, figuring out the resistance profile for the combination of antibiotics. And I will have to say that statistically, this is perhaps one of the weakest parts of the analysis because there are not as many studies reporting individual level isolate data that allows us to get at that correlation structure. Step 8 in this analysis was to estimate relative risks of death. The data on this are really quite sparse. We ended up with only 164 studies or data sources with only about 500,000 patients to allow us to get at the relative risk of death. So compared to the 50, 100 million plus types of data that we had in other aspects of the analysis, this is really quite a limited part of the work. And I see this as an area where more work, expansion of data collection, increased interest may really enhance the analysis. And then step 9 was to look at the literature and data sets on length of stay as the outcome and not just death. The last step, which is really accounting, is pulling it all together and then propagating the uncertainty in each of those steps into the final result using simulation methods or statistical sampling methods. And I only mention it because there's so much detail in the analysis of AMR that the computational load of doing this last step is actually quite substantial, creating petabytes and petabytes of data each time we run the whole system. Here's a summary of all the different types of data sources, in total about data on 470 million individuals, from multiple cause of death data, hospital discharge data, the microbiology, and a number of other sources, and how each of these different types of data feed into the 9 different analytic steps that I just walked through. So this sort of patchwork points out how important the sort of triangulation on AMR has become. And this is sort of our standard way of thinking about measurement or estimation in the GBD, because there isn't perfect data essentially anywhere that covers all the health outcomes that one might be concerned with. And in the case of AMR, where you have this cross-cut clinically across many different disease processes, it becomes even more important. I won't go through all the little numbers here, but here in the paper, we try to highlight that some parts of the world, for example, the high-income North America or Western Europe, we have quite good data on a large fraction of countries for each of those analytic steps. But there are some parts of the world, Central, Sub-Saharan Africa, where we have some data on the prevalence of resistance, but we have very little data on the other steps or analyses. So just by the nature of the data that are available, we are going to recognize that there is a considerable paucity of data in some low resource settings. Now, I do have to mention a diversion in the analysis that took us an extra sort of 6 months in the work, which was to try to convince ourselves and reviewers of the main article that the data are not overly skewed in low resource settings towards countries where we have only data coming from tertiary referral hospitals. This is particularly a concern for Sub-Saharan Africa. And so we did, for all the different sources, all the different hospitals that published studies, all the hospitals that we obtained data, we had to go back and classify those hospitals and other studies as to whether they were district hospital level or community-based hospital or referral hospitals, and then build that analysis in and try to correct for any bias there might be towards referral hospitals. Okay, enough on the methods. Let me go through some of the key findings. So at the highest level, about 5 million deaths a year in 2019 are associated with AMR. So for me, this was a much larger number than I expected to see. And I think really quite compelling as how common AMR has become around the world. Observation number 2 is that 1.25 million deaths are attributable in this much narrower definition of AMR, where the counterfactual is how much more mortality are we seeing in the world compared to if you had had a drug-sensitive infection. So truth is probably somewhere between these two numbers. Either way, whether 1.25 million deaths a year or 5 million deaths a year, it puts AMR amongst the leading causes in the world. And just for context, it's not an appropriate comparison, but it makes AMR, even with the narrower definition of 1.25 million deaths, in a similar magnitude, or actually somewhat larger, than HIV, TB, and malaria. Now, of course, they affect different age groups and different regions in the world, so that's probably not the best analysis, but it gives you some sense of just how big an issue AMR actually has become. On the right side here in the histogram is the death rate in the larger bar for each region from deaths associated with AMR, and the smaller bar is this attributable AMR. Either metric, it turns out that the highest numbers, which was a real surprise and partly why we went back and did this careful analysis of source by source, was highest in sub-Saharan Africa. This was not what we expected to see. And when we dug into this in great detail, you can understand this as having several components. First, the number of infections in sub-Saharan Africa per capita is much higher than in Western Europe, as an example. 4-fold, 5-fold higher pneumonia rates, as an example. And then, even though the prevalence of resistance is lower in Africa than in Western Europe, as an example, the death rate from AMR is higher because you have to take into account how much pneumonia there is, what is the case fatality of that pneumonia, and what is the prevalence of resistance. Now, when we look at drug-bug combinations, here we get this matrix. This is for death. We also have it for years of life lost. We also have it, in the appendices for other outcomes. And what we find is that there are certain drug-bug combinations, and this is focused on the narrower definition of attributable deaths, that the number one drug-bug cause of death in terms of attributable mortality is MRSA. And then we have a 6 other pathogen combinations that are between 50,000 and 100,000 deaths that are shown in orange. And those range from carbapenem-resistant Acinetobacter to fluoroquinolone-resistant or third-generation cephalosporin-resistant E. coli to Klebsiella, again for third-generation cephalosporins, as well as carbapenem, Mycobacterium tuberculosis, as examples of those categories. Now, if we look at it a somewhat different way, which is what are the infectious syndromes where there are the most deaths related to AMR, top of the list is pneumonia, then bloodstream infections, and then interestingly, intra-abdominal infections, and then you get into urinary tract infections, TB, and skin infections. Now, another thing that I think for this audience will not be a surprise, but is a huge surprise for most people that work on global health, and that is that there is a remarkable number of deaths in the world from a number of pathogens that have not had a lot of global attention. So if you look at ranked by drug resistant, the larger definition here, the associated with drug resistance deaths, top of the list is E. coli, then Staph aureus, then Klebsiella pneumonia, then Strep pneumo, Acinetobacter, Pseudomonas, then come in, and then Mycobacterium tuberculosis. There are a lot of people in global health that have not paid that much attention to some of these gram-negative pathogens like Acinetobacter or Pseudomonas. And so I think part of why we are separately writing up the analysis of deaths related to each pathogen, whether resistant or sensitive, is just to bring attention to the role of some of these gram-negative bacteria that have historically not had the attention that perhaps they should, because they cut across many different infectious syndromes, but they make for excellent vaccine or drug development targets. Now, there's another, the work is ongoing. And so what I'm showing here is, further analyses we're doing by the regional level and then by pathogen, and you'll see in a moment the time trends. And so this map on the top left is the pathogen responsible for the highest rate of attributable drug-resistant mortality. And there's quite a spatial variation in what is the number one AMR pathogen. And then the bottom right is an age pyramid going from youngest at the top to oldest at the bottom at the global level, showing the number of deaths by pathogen due to AMR across the globe and how much it varies by age group. Now, another way to try to communicate the results is to show some maps. And in the paper, we have maps both of the primary data on the top map and then the results of the statistical modeling exercise on the bottom that fills in gaps, deals with conflicting data, and deals with some small number issues. And this is the map for third-generation cephalosporin-resistant E. coli. This is the map for MDR, quite a different spatial pattern. When we go to third-generation cephalosporin, sorry, that's the repeat of the E. coli. When we go to carbapenem-resistant Klebsiella, you get, again, a very different spatial pattern. And part of the message in the paper is that it is not a single gradient from, let's say, high income to low income. There are these distinct spatial patterns in what is out there. I recommend, if you haven't seen it, the annex to the paper, which provides those sort of maps in detail for all the drug-bug combinations in the analysis. Some of them, there's much less data than for others. And so I think it's very important to just highlight that there are incredible gaps in the data. And the best way we can improve the assessment of AMR in the future is better primary data report collection and primary data reporting, because there's a lot of data that's out there in national monitoring systems that may not be getting into our analysis or even not being analyzed from a population epidemiological point of view, often because there isn't linkage between lab data and clinical data. And I think one of our main messages on improving data is the critical use and role of linked data that goes from the patient to the lab to the outcome. And so the other aspect and limitation of the study is, of course, this issue around the correlation structure of resistance and drug-bug combinations. Now, in the paper, like everybody else, we speculate a little bit, although it's not really driven by what is fundamentally a descriptive analysis on approaches for improving. And these are also become, reducing the burden of AMR. These have become targets for future analyses and collaboration. And I'm very happy to say that we have received funding, both the IHME side and the University of Oxford, for a second phase. The primary funder was the Department of Health and Social Care in the UK, but also Wellcome Trust and the Gates Foundation. And certainly the first two have now decided to fund a second wave of the analysis. And so that's where we will be looking at the historical time trends so that we can then also forecast in a credible way where AMR may be getting worse. Now, one of the other obvious conclusions from looking at these results is the critical role of R&D in creating new drugs. And I'm happy to say that this paper seems to have captured the attention of some of those companies, and that's helping them prioritize new research. Now, with an eye on the clock, let me end with this slide and just say that this is a live undertaking. It's an active collaboration. It's a collaboration we welcome others to join. And the key things that we're addressing now is trying to get the time series back to 1990, to release the results at the country and subnational level, to make all these results available to any researcher and anybody else online and in a visualization tool, and to move which is the goal of the second wave of research from that's being funded, to routine integration of measuring the burden of AMR into the regular GBD cycle. Thank you very much for your attention. Sorry for running over 2 or 3 minutes. Okay.
Thank you very much, Chris, for a wonderful, comprehensive and informative lecture. And we are all looking very much forward to future results to come. And again, let me say, you know exactly how much you how much stress you also put on political decision makers by publishing this data. And I'm very thankful to you also for this. Have a good day. We will enjoy a party now after this fantastic talk. I hope you can enjoy your day also. Bye-bye, Chris. We will send the certificate by post, by shipment.
Thank you so much. Bye-bye.